I'm Pete Allcroft. I'm a respiratory and palliative care physician from South Australia and work at Flinders Medical Centre, and have been involved with motor neuron disease clinics since about 2000, and work closely now with David Schultz, who joined us about 6-7 years ago. So I'm chairing this afternoon's session, and it's about trials and patient and client engagement. And if one thing I get asked all the time by our patients is what trials are on, what can we get access to? And in the middle of Australia, we've been hamstrung because it's expensive to bring trials to small states. So I'm really excited to hear what's available, what's coming up. Before I start, you will have seen as you entered your row some questionnaires, an evaluation form, and the MND New South Wales is keen for you to provide feedback on what you've enjoyed today, what you feel we've missed out on, how we can do it better for next year. So if you could fill those out and leave it at the registration desk when you leave, that would be greatly appreciated. The first speaker this afternoon really needs no introduction, Professor Matthew Kiernan. He's visited South Australia twice this year to speak with us, and he is a very generous person. This is his alma mater. I'll be short. He's a clinician, he's an academic, he's a researcher, he is a true advocate for motor neuron disease, and I'd like to welcome him to the podium to present.
Thanks, Peter, for your very kind introduction. In fact, the stuff that you have done and organised in South Australia has been very impressive and appreciated, and certainly the patients have spoken very highly of your service, so it's great to have you visiting us here in Sydney. So look, I think I'll obviously going to be focusing on mostly the present and the future, but I will just mention a little bit about the past. So there's various ideas about what motor neuron disease is, but probably the most prevalent theory is excitotoxicity. So the motor neuron is triggered off and it's repetitively firing. It releases glutamate, and glutamate is toxic to motor neurons, and that causes the degeneration and death of motor neurons. And that theory came from colleagues, particularly Pam Shaw in the United Kingdom and Jeff Rothstein in America, and they started measuring the cerebrospinal fluid of patients with motor neuron disease and found that glutamate was increased. And I think from that, the first positive treatment was riluzole in motor neuron disease. This was published in the New England Journal of Medicine and also in the Lancet, and we were part of those studies in the early and mid-1990s, and we were able to eventually, after a lot of political discussions, get riluzole introduced as a treatment here in Australia. I think one of the other key discoveries that we've mentioned is the idea, the linking with superoxide dismutase, and that was a key discovery, and that led to a whole area of molecular understanding of motor neuron disease and the development of SOD1 transgenic mice models. And that's been, I think, critical in terms of understanding patterns of degeneration and looking for targets of therapies. So whether it's the supporting cells of the motor neuron, the astrocytes, whether it's glutamate, obviously how the genetic mutations lead to problems with transport of proteins in axons and the release of messages from the nerve to the muscle. But I suppose there's always a downside to discoveries, and one of the questions that is out there now is how useful has this model been for what we understand is the human disease, motor neuron disease. And this might seem like a controversial thing to say, but we've had various debates at international symposium about the Sub1 mouse. And why would I say that it hasn't been great? Well, it's now about 170 positive trials in the Sub1 mouse model, and not one of them have developed into a treatment for human motor neuron disease. And in fact, the riluzole study was done in patients first, and then it was trialled later on, once it was already positive in patients in the mouse model. So why are the trials always negative that have come from the Sub1 mouse? Well, firstly, treatments are started in mouse models typically once the mouse is born, so in litters. And for us as clinicians, we only see patients once they've developed motor neurone disease, and that can be typically in their 40s, 50s, 60s, 70s, but we don't know what's been going on for the 30 or 40 years beforehand, so we can't start that treatment. The other thing is that, and this is, I suppose, something that's come out more recently, is that SOD1 is an unusual form of motor neurone disease. So all other forms of motor neurone disease have a protein inclusion in the brain and the spinal cord, which is TDP43, and SOD1 doesn't have TDP43. And is not typically associated with cognitive changes, and is not typically associated with frontotemporal dementia. So SOD1 really, I think it was great because it put motor neurone disease back into people thinking. It's worked out plans of degeneration, but it hasn't been useful for patients with the disease. And I think we really do need to get communities better organised in large groups, and to be able to undertake proper sort of large-scale clinical trials. And with every clinical trial, there should be biomarkers linked to the trial, so that even if the trial is negative, we get some understanding of the biology of the disease in patients. And obviously, we try to set this information through, and this is the Australian Motor Neurone Disease Registry, which was established by Paul Talman, but involving representatives from each state in Australia. So in terms of where we're at now at the moment, and I know that Julian is going to follow me, so I won't go too much into this, but this is the Lighthouse project, and I suppose it's important to say that it was jointly funded by the Motor Neurone Disease Research Institute of Australia and the Cure for MND, or Fight MND now, Foundation. And I suppose HIV is one of the examples of medicine where it has been an incredible triumph. So going from, universally catastrophic and death sentence to patients now dying from other causes and having HIV for decades. The other positive advantage of that is anyone who specialised in HIV medicine has nothing to do now because they've been so successful in their treatments. And so they're bouncing around to other fields of medicine, and Julian has looked across the landscape and he's realised that motor neurone disease has a clear need, and so he's bringing that expertise for us. And we heard in the last talk about the upregulation of inflammation in motor neurons. So when motor neurons are dying, other cells come in to basically clean up debris, and that's these inflammatory cells. So can we try and come up with an inflammatory approach? And this treatment is Trimec. Julian will go into it. Suffice to say that it's a study that enrolled patients in New South Wales and Victoria, 4 sites. It's completed, and Julian will present the results next. Another study that is currently underway it's been driven from Australia. So a compound called Seaway TSM, which was initially discovered as a way of imaging the brain, a ligand in a metabolic PET study of the brain. And it also has a way of increasing copper in the brain. And the Florey in Melbourne have the patent on this. And it went around globally, and a company here in the United States called Collaborative Medicinal Development, CDC, came to Australia and looked for funding, and in fact we were able to introduce them to Babcock and Brown. So they were the investment bankers. Jim Babcock, who lives in Sydney, and he has bought into it and is funding, in conjunction with other philanthropists, this study. At this stage, the Seaway TSM study has been a dose-defining series of studies, so putting 3 patients on and just dialing up the dose to see what is the toxic dose. And those studies are finished, and now it'll be a phase 2 study once the best dose is worked out, which will be done in the next few months. The results from this study will be presented at the International ALS Symposium in Boston in December. In terms of other therapies that we know are important, and I'll get onto some more clinical trials in a moment, one thing we do know is management in a multidisciplinary care setting improves outcomes for patients. So this was our original team and a number of people here. So Margie, the clinical nurse consultant, is up at the back row there. And I suppose the most important thing is case management of patients. And I think this is something that we'll need to tackle with the NDIS, but it hasn't been worked out in any proper way at this stage. But a multidisciplinary clinic incorporates representatives from the MND Association, occupational therapist, a speech pathologist. This is Whitney, the physiotherapist, who's still here up the back, waving. And a social worker, palliative care physician, and another physiotherapist. And we also have a dedicated respiratory physician and a gastroenterologist. And this is data that came from Orla Hardiman comparing patients who are managed in a multidisciplinary clinic versus just standard care from a neurologist. And that increases 50% survival of the order of 300 to 400 days. So an extra year in milder forms of the disease by just attending a multidisciplinary clinic. Obviously, we have a lot of symptomatic therapies, and Peter will be more of an expert, but in terms of, we can treat a lot of the symptoms that patients are experiencing. And we also heard earlier about non-invasive ventilation, also the key message about avoiding weight loss and caloric, increasing caloric intake and so on. And all of these, again, are relatively proven in motor neurone disease. So another company is Flex Pharma, that's United States based in Boston, and they had a new therapy for treatments, particularly for cramps and muscle twitching, fasciculation, and this was run again in Australia. So the Australian government has put money on the table to bring companies to Australia to run trials and give them more income and promote them. So that's why more companies now are coming to Australia. This study has now been completed and again will be presented in Boston, but I have spoken to the principal investigator. We're one of the sites, and it's been a positive study in terms of better control of pain, muscle cramps and spasticity. So this is a medication, a monoclonal antibody, masitinib, which is from a French company called AB Science, and they presented their preliminary results at the European ALS meeting earlier this year. And this is a treatment which is an inhibitor of tyrosine kinase, which is one of the pathways that are involved in motor neuron degeneration. And so this was released earlier this year, saying that they had positive results from a phase 2 study. They now want to do a larger phase 3, so multi-sites around the world. We'll be one of the sites, and we're trying to get more Australian sites enrolled, and this will likely be during next year. At the moment, they're just working out budgets, but even once you work out budgets, there's a delay in terms of getting the study protocol, putting it through an ethics committee, and then getting all the medication on sites and so on. So this will probably be another 6 to 12 months away. So the other big news is edaravone, which is owned by Mitsubishi Pharma, and again, working on this sort of excitotoxicity. Superoxide dismutase is also an enzyme that's involved in scavenging of free radicals, so it's the same sort of concept of a pathway. This was a treatment that was used in stroke, so it's standard care for stroke in Japan. It reduces the tissue damage around the area of stroke. And having been state-of-the-art therapy for stroke, they wanted to try other disease states, and they've done a number of trials now, Mitsubishi, in motor neurone disease. And their studies, they hadn't released all of the data until very recently because they wanted to put all the data to the Food and Drug Authority, FDA in America, and they just got approval from the FDA for edaravone as a treatment for motor neurone disease. And I suppose the key message is that it's for younger patients, and this is a respiratory measure, forced vital capacity greater than 80%. So basically, it should be patients right at the beginning of the disease course. And there's been a number of approaches to look at the population who attend clinics, and probably this would be about 7% of the total range of motor neurone disease patients. Now, a number of Australian patients have gone to Japan. I should say that it's very invasive, so it's 10 to 12 infusions per month for 3 months. And so they've been getting the treatment in Japan, and then they brought edaravone back to Australia, and some institutions have been able to give it on a compassionate basis. So we're currently looking at funding sources and also, again, applications to the pharmaceutical benefit scheme. That will be some period of time. Another study that has recently approached Australia for involvement is Saito Kinetics. So they did a study, and this is a way of stabilising the neuromuscular junction where the nerve goes to the muscle. And in their initial studies, which probably didn't have enough patients, one thing they did show was there is improvement in respiratory function. And so they wanted to do a trial just based around respiratory function and reserve in motor neurone disease. There are another family of medications that stabilise the membrane of the motor neurone, and these are being undertaken in the United States at the moment by the Northeast ALS Consortium, the NEALS Consortium. There are 14 sites in America doing these studies. We are the data centre for all of their studies, so they're sending their data to us, and I think there'll be some early results coming out probably next year. Patients always ask about stem cell therapy, and this is something that we're all keen to try and develop as well, regenerative medicine. It's probably unlikely that we could rebuild the whole nervous system in patients with motor neurone disease, but if you inject stem cells in the right area, can you preserve the life of a motor neuron or make it function better? And Brainstorm are based in Israel and Boston, and we've made various approaches to them to set up a site in Australia. They, again, are very focused on getting FDA approval, and they don't want to slow that down by involving Australia. So some patients have gone on to Israel and America and had treatment. It's obviously that you have to pay for the full service, which is very expensive. But that's something that we will have more discussions with them at the Boston meeting, and there's certainly been a significant philanthropic support from Australian patients and their families, and they've said that, they've put 10 million on the table to bring Brainstorm to Australia. We haven't been able to get them to do that as yet. In terms of the future, look, I suppose what I'd say is that motor neurone disease is a lot of diseases that are put together, and we're now starting to understand that all things aren't equal. And there was a study that was published in Neurology last week, an American journal, and they looked at the lithium trial, and they showed that even though the trial was universally negative, if they they looked at patients who had a specific mutation in their genes called UNC13, that those patients actually benefited from lithium. So now I think even though we've had a lot of negative trials, we need to go back understanding the genetic changes and see whether we can dissect this better. Maybe some of the negative treatments may have a role in certain forms of motor neurone disease. The other thing is that most patients can say where their motor neurone disease began. They don't sort of come along and say that it was a global change, like all their arms and legs and everything was affected. So they can usually say it's like maybe my right hand or my left foot or my speech. And I think that's part of the message. I think if we can try and get appropriate treatments into the areas that supply them, and when I say areas, this would be brain areas. So I think we need to focus on the motor strip in the brain and inject or get appropriate treatments in there. That's where we're probably going to have the best chance of success. The other thing to say is that this is a fraud area, and it relies on we heard earlier about the first in humans, phase 1 and phase 2. But if you make mistakes in that area, it leads to real catastrophes. So we're involved in this dexpramipexole study that was 990 patients from Biogen. It was a $40 million trial that was negative, and a lot of Australian patients were involved. And going back to the original phase 2 study, the reason why it didn't work was the data at the phase 2 stage was not correct. So the other thing I would say is this is where we see patients at the moment. Once they've presented with weakness and they're getting progressively weaker. But we don't really know what has gone on from the period of basically conception, pregnancy, early growth, development, And this is where the real focus is now. So if we could identify a patient 5, 10, 15 years before they developed motor neurone disease, you could see that we'd have a much better chance of protecting their nerves and neurons. And that's not too fanciful. So for instance, in Parkinson's disease, the partners of patients with Parkinson's disease say they have noticed sleeping problems, disturbed sleep, rapid movements of their legs 4, 5 or 6 years before they develop Parkinson's disease. And I think there's a great focus now on personality, cognitive changes, various forms of imaging of the brain. But I think also in some individuals who have genetic mutations linked to the disease who don't have the disease, but also in general population, large-scale approaches. And I think that's where we'll get some further information. And I think the other thing I would say is there's a lot of interest in motor neurone disease. There's a lot of, we heard about the Ice Bucket Challenge, but also pharma now are investing very heavily in motor neurone disease. And we're seeing some of the fruits. So in 1993, riluzole, this year was the second medication to get FDA approval, so 93 to 2017, but I think there's about 3 or 4 that are very close to being on the cusp, and I think we'll be able to eventually be able to say, well, there's 4 or 5 treatments, and maybe this is the best first-line treatment, then add this and this medication, and I think, that it's a lot more promising than it would have been 5 years ago. So I might finish at that stage. Thank you.